Okay, welcome to Let's Get to the Marks. This is an A-level biology walkthrough on some questions on the synapse. What I suggest you do, pause the video, pen, paper. You won't need a calculator for this one, just pen and paper. Have a go at the question, write down the possible answers, and then unpause and watch me go through with the explanation. All right, let's look at the first question here we got in this little walkthrough pack. The figure below shows a drawing of a neuromuscular junction. So, name the parts A to D. So why don't you press pause, have a go, or I'm going to go through the answers. So the first one here, you should be able to tell we've got cristae inside. This is that organelle, mitochondria. So you can say mitochondrion, which is the singular, or mitochondria. They'll accept both. Now let's look at B. It's the membrane around here. Now you can call this the synaptic knob, no diddy, or you can call it the presynaptic membrane. Personally, I call it the presynaptic membrane, and that is going to be at the end of your sensory neuron, your motor neuron. In fact, this is a neuromuscular junction, so this is going to be the motor neuron where it meets the sarcolemma. So let's go. What is C? C, what are these little circles? So you might be thinking, is it neurotransmitter? Is it vesicles? Well, these are those little sacs, often drawn bigger than this. These are the vesicles, the ones that are stimulated to move to the presynaptic membrane when calcium channels open, when an action potential arrives, and the calcium ions cause these to fuse and release their neurotransmitter into here by exocytosis. So the answer for C, enough whiffle waffle waffle of mine, is vesicles. And then there's one possible answer for D. Now, the most common mistake here is people will say the synapse. Well, the synapse is the whole thing, the presynaptic knob, the postsynaptic membrane. In this case, it's at a neuromuscular junction, so it's the sarcolemma. What is the specific name for that gap there? You can call it the synaptic gap, but the proper name is synaptic cleft. Now, you might say, how's this only 2 marks and not 4? Well, when they do this type of thing, you need to get 2 for 1 mark and 2 for the second mark. There we go. Hopefully you did well on that. Let's move on to the next question. Right, let's go. Following the release of acetylcholine into a neuromuscular junction, a muscle contraction occurs. Describe the sequence of events. Sorry, that was a muscle contraction. I wasn't doing my pants. Describe the sequence of events following the release of acetylcholine that leads to the stimulation of this contraction. Do not include in your answer the events following the release of calcium ions in the myofibril. So they've been really particular here. They want the events following the release of acetylcholine. So they don't want you to talk about calcium ions causing the vesicles to fuse, exocytosis. They don't want none of that. They just want from once the acetylcholine enters the cleft, what happens until calcium ions enter the myofibril. So why don't you pause the video? It's worth 4 marks. So try to make 4 separate bullet points that you can think of. Have a go. Right, I'm going to go through the answers now. So here is my presynaptic membrane. This is going to be the synaptic knob, the presynaptic knob of the motor neuron, because we're at a neuromuscular junction. We're going to have vesicles. This has all happened before we start our answer. They want to know what happens after. This is my little triangle. That's my acetylcholine. They want to know what happens after that is released into the synaptic cleft. Okay, so we've got our acetylcholine being released into the synaptic cleft. So how are you going to describe what happens next? So the first marking point, acetylcholine binds 2 receptors on the sarcolemma. Or maybe even we need to say that how does acetylcholine, how does it actually cross the synaptic cleft? In fact, I think that's a better thing to say. So I would say acetylcholine diffuses across the synaptic cleft, first of all. Keyword I've put in bold there. You don't want to say it moves across. You're going to get a mark for saying acetylcholine diffuses across the cleft. Then we want to say acetylcholine binds to receptors on the sarcolemma. They will allow you to say the postsynaptic membrane, which is the generalized term, whether you're dealing with a sensory to relay or relay to motor, or in this case, motor to neuromuscular junction. But specifically at the neuromuscular junction, you can call it the sarcolemma or the cell membrane or the membrane on the sarcolemma. So the third point is that this binding of the acetylcholine to these receptors, if enough acetylcholine binds, we're going to get sodium gates opening, and of course if enough sodium ions enter, we're going to cross the threshold, we're going to get an action potential forming. So the next point is that sodium ions move in through sodium channels that open. So sodium ion channels open, sodium ions diffuse in, this facilitated diffusion obviously, and depolarize the membrane of the sarcolemma. Now very key that you don't say sodium diffuses in, because they want the word ions, because sodium atoms aren't charged, so they're not going to cause depolarization. So if you just say sodium diffuses in, they're going to say, well, that wouldn't cause depolarization. So the reason it has to be ion in A-level biology is because you need a positive ion to move in to cause that plus 30 millivolt, that depolarization of the membrane. All right, now the fourth mark, once that membrane is depolarized, what does that do? Well, that causes the sarcoplasmic reticulum to release calcium ions, which will eventually work their way into the myofibril. We'll get the whole troponin, tropomyosin stuff, which the calcium ions do, the whole heads and all of that business. But we don't need to talk about that. The fourth mark is just calcium ions are released from the sarcoplasmic reticulum. Now, you can call it the endoplasmic reticulum, so it's very similar. Check out my GCSE level D drawing on that one. This is going to get you to 4 marks. So if you don't know this, make a note of it. All right, let's move on. Inhibitory synapses cause hyperpolarization in the postsynaptic neurons. Explain how this inhibits synaptic transmission. It's worth 2 marks. So how does hyperpolarization stop an action potential forming in the postsynaptic neuron? Explain. Have a go. Pause. All right, I'm going to go through the answer. So I'm going to give you an example here. So for example, I could have a couple of these presynaptic knobs meeting with my postsynaptic membrane. Now, maybe it's acetylcholine. This one is releasing here. And normally, under normal circumstances, this acetylcholine is going to bind to these receptors. And if enough of it binds, enough sodium gates will open that an influx of sodium ions will polarize this membrane in the postsynaptic membrane, and we'll get an action potential formed here. Now, how can that be inhibited? Well, we can have inhibitory neurotransmitters released from an inhibitory synapse, and what they're going to do, these neurotransmitters, whatever it's called, who knows, it could, just as an example, it could cause chloride ion channels to open and chloride ions to diffuse into the membrane. Now, instead of this being minus 65, what can happen there when negative ions diffuse into the postsynaptic membrane? It can make the resting potential more negative. Now, it's very key that you don't say reduce the resting potential. The resting potential is going to get bigger, right? If it gets more negative, then the difference between it and the outside is larger. Okay, so don't say it reduces it. It can make it more negative, i.e., instead of being minus 65, it could go to minus 100, let's say, or minus 120 millivolts, hyperpolarized, much more negative. Okay, now this hyperpolarization, remember, you normally have a threshold potential around minus 55. So let's say it's minus 65, minus 70. Normally, we just have to swing that by about 15 millivolts. We just have to swing it from minus 65 to minus 55 or minus 70 to minus 55. Once that happens, we get that positive feedback and all those sodium ion channels open, sodium ions influx in. Now, if it's gone to a minus 100 millivolts before the, it's much less likely to be depolarized, basically. It's less likely to be able to cross that minus 55 threshold. You'd need a swing of 45 millivolts for it to cross that minus 55 threshold and that positive feedback to happen and all those sodium gates to open. Anywho, for the marking points, you just want to say negative ions diffuse into the postsynaptic membrane, making it more negative, less likely for an action potential to form or less likely for depolarization to occur. Right. Welcome to Let's Get to the Marks, going through question 2. This is an A-level biology question on the muscles from the 2024 paper 2 for AQA. 2.1, figure 2 shows part of a relaxed myofibril. Length of the sarcomere is 2 micrometers. The myofibril represented in figure 2 has 34 sarcomeres. The length of every sarcomere is 2 micrometers. After contraction of this myofibril, the length of each sarcomere changed by 20%. Use the information provided to calculate the length of the myofibril after contraction. Give your answer in millimeter and in standard form. Okay, we know there's 34 sarcomeres in one myofibril. Of course, they're made up of myofilaments, actin, and myosin. Those are the lines on the diagram, but we're looking at the whole length of the myofibril. There are 34 individual sarcomeres. Each one is 2 micrometers in length, so we're going to do 34 times 2, gives us 68 micrometers. I swear my little dodgy micrometer sign is well dodged. Now, after contraction, what's going to happen to that length? If you think about when muscles contract, what happens? They get shorter. So when you squeeze your muscle, the myofilaments slide over each other. Remember, you have the myosin heads inside those actin, and they slide across each other. So it's gone down by 20%, so I'm just going to times it by 80%. You could do this all sorts of ways, but basically reduce 68 by 20%. So you choose the way you want to do it. 10% is 6.8, so you could do that, whatever. I just times it by zero point 0.8. So we get 54.4 micrometers. We need to turn it into millimeters. And we need to put it in standard form. So divide by 1,000, you're going to get 5.44 times 10 to the minus 2. If you really want to know what that is, normally it is 0.0544 millimeters. But of course, they want it in standard form, and that's going to get us the marks. Okay, let's move on to 2.2. What is 2.2? Which statement correctly describes the changes in length in a sarcomere when a myofibril contracts? So we got 4 different possible answers. You've got to tick one box. So the H zone decreases, the I band increases, the A band decreases, the H zone increases, I band decreases, A band decreases, the H zone decreases. Oh, you get the idea, right? There's all these different options. You've got to decide which one is the best. You should learn this off by heart, actually, during a contraction, what happens, and then you can just slam that down. You don't even have to look at it. But if I was to go through this, what I would talk about, excuse my funny sounding voice, I've got a dirty cold at the moment. Essentially, you've got 3 areas here on the sarcomere. You've got the H zone. Now, the H zone only contains that thick black line. You see the thick black line there? That is myosin. I've drawn those myosin heads in pink, and they slot in to the thin line, the thin myofilament called actin. Now, obviously, the length of each of these lines does not change. But what happens is they overlap more, hence the term sliding filament theory. So those actins are going to slide over the myosin. So the region that is just myosin is going to become smaller. So our H zone decreases. Now, when we look at the I band is the bit that is actin only. So it can only have actin. Okay. And so the I band is also going to get smaller. Just look at those. If you slide the thin lines over the fat lines, the bit that is only thin is going to get smaller. So the I band gets shorter or decreases. Now, if we look at the A band, very similar to the H zone, it corresponds exactly to the length of myosin. Okay, so the thick black band. So it is the length of the myosin myofilament. And, but it can be overlapped as well. Now, essentially, the length of the myosin filament, oh my gosh, yes, it's not going to change. Okay, it's not just myosin on its own. So the length of the A band is not going to change because it's wherever you have that thick line. It doesn't matter if it's overlapping. So the A band will never change its length, whether contracted or relaxed. So really, you need to know during a muscle contraction, H zone shrinks because it is the region with only myosin. I band shrinks because it's the region with only actin and the filaments have slid over each other. And the A band stays the same. There's no change in the A band. And that is because it corresponds exactly to the length of the myosin thick filament, but it doesn't matter if it overlaps. Okay, let's go to 2.3. That was long. Fast muscle fibers have a higher concentration of glycogen than slow muscle fibers. Explain how the difference in glycogen concentration is related to the different properties of these muscle fibers. You might want to pause the video, grab a pen. It's 4 marks. So, why do your fast twitch muscle fibers have more glycogen? Well, fast twitch muscle fibers really get activated when you're like doing a tennis serve or when you're lifting some weights, you're trying to deadlift a weight or squat a big weight in the gym, or when you're throwing a punch, or any sort of movement, lifting your chair, lifting your desk, any sort of movement that's intense over a short period of time, like a tennis serve, is going to require you to use your fast twitch muscle fibers. So why do they have more glycogen? So the reason they have more glycogen is because the fast twitch muscle fibers can convert the glycogen to glucose, and they do this by a process called glycogenolysis. Oh my gosh. Yeah, hard one to remember that. Glyco, meaning glycogen, glycogen, and then they just randomly put an O in. Glycogen lysis would be better, but it's glycogen-o-lysis. Glycogen-o-lysis? Yeah, anyway, the glycogen breaks down to form lots of glucose. Now, why would you need lots of glucose? This means you can rapidly produce energy by anaerobic respiration. There's no need to wait for oxygen to be delivered by the blood to the muscles. You can produce energy straight away. Now, it's only a small amount of energy because it's going to be anaerobic respiration. Okay, and you're only going to make a small amount of ATP, but it's that little bit of extra energy you need. So Fast twitch muscle fibers have lots of glycogen, so they can break it down to form glucose for anaerobic respiration to produce a small amount of ATP in a short amount of time. This happens rapidly or quickly. We want to put the time there as well. This is rapid or fast. I've used the word rapidly. Nice. Right. Whereas your slow twitch muscle fibers, these do not need as much glycogen because they're predominantly used for aerobic respiration. They're going to produce a lot more energy because aerobic respiration produces a large amount of ATP, but it does it over a longer period of time. So you're going to rely on your slow twitch fibers more for those endurance exercises. But it does produce a lot more ATP, but it's slower over a longer amount of time. And you're kind of born with different amounts of those fibers. I think there's various research either way about how much you can adjust the type of fibers genetically that you have. Like you might be someone who's more fast twitch, someone who's more slow twitch. Hence, you might be more suited to the 100 meters or more suited to the long distance running. You know that from PE. People are either or, but you can change them a little bit. Anyway, enough of that. Let's move on. How you going to do it if you really don't want to revise? Get down on it. Come on. Right. Welcome to Let's Get to the Marks. Going through this A-level biology question on photosynthesis. So let's get into it. What you should do is watch me read through the question, then pause the video, have a go on a scrap bit of paper, and then unpause and watch my explanation. All right, let's go, let's go, let's go. Biologists investigated photosynthesis, 3.1 this is, measured the percentage of light absorbed by a plant when it was exposed to different wavelengths. Let's read that again a little bit more properly. A biologist investigated photosynthesis. They measured the percentage of light absorbed by a plant when it was exposed to different wavelengths. They measured the rate of photosynthesis at each wavelength of light. Figure 3 shows the results they obtained. Here's figure 3. We can see rate of photosynthesis is shown by this dashed line. So we can see it's going up, down, up. It's like an M shape. And then the percentage of light absorbed is shown by this solid black line. And we can see it's matching that dashed line. So there is definitely some sort of link there, right? And then at the bottom here on the x-axis, we have the wavelength of light in nanometers. So 400. It's a short wavelength, high frequency, high energy, which is blue light. Up to 500. Then we've got green light between 500 and 600, and then red light, which has a larger wavelength, slightly less energetic red light. Using figure 3, what can you conclude about the relationship between the percentage of light absorbed and the rate of photosynthesis, the color of the light and the rate of photosynthesis? Now, this is worth 2 marks, and they're just asking you for a conclusion. So say what you see. Don't try and explain anything. Just say what you see. It's worth 2 marks, so it's going to be 1 mark for each. The percentage of light absorbed and the rate, how are they affecting each other? The color of the light and the rate, how are they affecting each other? Why don't you pause the video, have a go at this one, and then unpause and watch the explanation. All right, I'm going to go through it. So let's have a little look. We can see that the percentage of light absorbed is the solid black line. And as this goes up, so does the rate, the dashed line of photosynthesis. As it goes down, so does the dashed line, which shows us that the percentage of light absorbed has a positive correlation with the rate of photosynthesis. I don't want to say they're directly proportional because that would mean they go up in exactly the same pattern, and it's not perfect, is it? But roughly, they go up together, they go down together. So we're going to say there is a positive correlation between the percentage of light absorbed and the rate of photosynthesis. Now, this is just a conclusion. So this is all you're saying. You're not explaining why. You're not saying, oh, the more light, the more, whatever, more light absorbed by chlorophyll, the more this and that, blah, blah, blah. You're not bussing out any science. You're just saying what you see with a conclusion. Just remember that. Right, the color of the light and the rate of photosynthesis. What do we notice about the colors of the light? We've got 3 light colors, blue, green, and red. We need to mention each of these to get this second mark. So see if you can do it. So I would say that the rate of photosynthesis is high with the blue light. You could give, if you want, you could say with blue light at, what is that, 475 nanometers, it's high. With green light, it looks like the rate of photosynthesis is low, and then with red light, it is also high. So I'm going to combine blue and red together in a sentence and say the rate of photosynthesis is high with blue and red light. The rate of photosyn, oh my gosh, synthesis is high with red and blue light absorption. I can't spell, but we're not going to worry about my spelling for now. Oh my days! I need to make that smaller. You can't even see that. That's so ugly. Now, one thing that you'll see on the mark scheme here is that will not get you the mark alone. You must mention all three colors to get the mark. So we're going to say the rate is low, and they can assume because I've mentioned rate of photosynthesis that I'm talking about the rate of photosynthesis. Don't want to write that again. The rate is low with green light. So I also want to make that to get the mark. You need to mention all three colors here to get the mark. So the rate is high, or photosynthesis is high with red and blue light, and it is low with green light. And that will be shown in the mark scheme by them writing a word in bold like and it means you need both points to get the mark. So always look out when you're studying a mark scheme for the bold words, because they mean you must have it in the answer to score the mark. Okay, let's move on. Ba-da-ba-da-bum-bum-bum. Right, what we doing? Oh, one more page. Lovely bubbly. 3.2, give three environmental factors that should be controlled when measuring the rate of photosynthesis in this investigation. Do not include features of the plant in your answer. It's worth 2 marks. You're going to need at least 2 of these to get 1 mark, and all 3 to get both marks would be my guess, because there's 3 points, 2 marks. It says don't include features of the plant, so don't say species of plant, surface area of the leaves. Don't do any of that. Nothing about the plant itself. So we're really looking at abiotic factors, peeps. Pause the video, have a go. All right, I'm going to go through the answers. So 3 environmental abiotic factors that should be controlled. So let's think about what are the limiting factors of photosynthesis, and that will get you all your marks, right? So we're going to say the carbon dioxide concentration. So we want to make sure that each plant, each time you do it, they have the same amount of carbon dioxide, because that will affect the rate of photosynthesis. So I'm going to say the carbon dioxide concentration. I also want to think of another limiting factor, temperature, of course. So the temperature, we don't have to explain it. We're just stating we're giving these factors. So the temperature should be the same. Now, of course, that's to do with enzymes. If it's too high, it could denature the enzymes. And we know between sort of 10 and 30 degrees, with every 10-degree rise, there's a doubling in the rate of reaction. So you want to keep that the same, obviously. And what's another limiting factor? We've got carbon dioxide, temperature. They might allow you to say water, even though that's not one of our limiting factors. Now, light. Now, we know they were changing the color of the light, but what is it about the light you should keep the same? They're changing the color, the red, blue, green, but you want to keep the intensity, which means the amount of light, the same, the power of the light. Like, so if you're using a red light bulb, blue light bulb, green light bulb, you want to keep use a 60-watt bulb each time, the power. So I'm going to say keep the light intensity, the same each time. Okay. And that's going to get you the 2 marks. And I put 3 ticks, but it's going to get you 2 marks. You might be able to mention H2O, but it is sometimes a limiting factor. It's not one of our main limiting factors, but you might be allowed to mention the amount of water there. Okay, it is one of the reactants in the photosynthesis equation, as we know. The water supplies hydrogen ions to the electron transport chain, chemiosmosis, so it is important in that light-dependent reaction. Right, describe how the products of the light-dependent reaction are used in the light-independent reaction to produce triose phosphate, which is TP. So do not include the role of ribulose biphosphate, RUBP, in your answer, and this is worth 3 marks. So we want to think about the light-independent reaction, which is what we're talking about, the Calvin cycle. How is it that the products of the light-dependent reaction affect the Calvin cycle? Explain that for 3 fat marks. Pause the video, have a go. All right, I'm going to go through the answers. So obviously you've got your CO2 coming in, right? Like In fact, I'm not going to do that over here. I'm going to do that on the side over here. This kind of long ass, you might want to forward through this bit, but it's a bit long, but you've got your carbon dioxide coming in, right? Now, what does that bind with? That binds with the RUBP. This is Rubisco. Check out the handwriting. What? Serious styly. Um, oh, oh my gosh. Yeah, that joins with RUBP. Now, they said don't mention this, so this will get you no marks. I actually don't know why I did it. What a plum, right? And that's going to give you two GPs that are 3 carbon each. So, if you know your Calvin cycle off by heart, you're going to get all the marks here, right? This is the bit we're talking about. What products from the light dependent reaction are needed to turn GP into TP? So this is the bit we're focusing on. And they go from glycerate, 3-phosphate, whatever, to trio, 2 triose phosphate molecules. And what we need from the light dependent reaction, what comes down from our stroma, um, from our thylakoid membrane, sorry, what comes down from our thylakoid membrane. Remember the light independent reaction. This reaction is happening in the stroma, the kind of Calvin cycle, and on the thylakoid membrane where we have chemiosmosis, that's where we're going to make ATP, and the ATP, of course, is going to be turned into what? It's going to be turned into ADP, excuse the crusty handwriting because it should be capitals, plus inorganic phosphate. And you use 2 ATPs and they become 2 ADPs plus inorganic phosphate. And then I'm going to attempt to write it 2 NADPH. Now, they prefer in AQA to say NADP reduced. So on the mark scheme, that will be the preferable thing. And the reason is because sometimes NADP can be NADPH or NADPH2. So they prefer you to just say NADP reduced. When you have hydrogen attached, you are reduced, and that will become NADP. Now, what happens to the hydrogen that is lost here? Well, it joins the GP, and when hydrogen joins something, it becomes reduced. So what we want to say in our answer, which we're going to construct now, our 3-mark answer based on that diagram, we want to say the GP is reduced to TP, okay? And what is it reduced to TP by? By Na, whoa, oh, my days, by NADP reduced, which loses hydrogen ions, which are gained by GP to become TP. I think I've longed that out. The other thing is energy for this reduction is provided by ATP, which becomes ADP plus PI. And so that's providing the energy for the reaction. The NADP reduced is providing the hydrogen ions, and together they reduce the GP to TP. It's a reduction reaction, and that's going to get you all of the fat marks. And by the way, the ATP and the NADP reduced, they both come from the light dependent reaction. It's the whole purpose of the chemiosmosis is to make NADP reduced and ATP. Of course, there is a third product that is made, but that is oxygen, which is the waste gas that's released from the stomata. Okay, so that's not included in the light independent reaction. That's not its function. Okay, that's the end of the question. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. Let's smash the exams. Welcome to Let's Get to the Marks, going through a Hardy-Weinberg calculation here for A-level biology plus dihybrid inheritance. Let's get into it. Why don't you pause the video, have a go at these questions, and then unpause, and then I'll go through these first 2 questions, 4.1 and 4.2. So in fruit flies, a gene for body color has a dominant allele for gray body, that's capital G, and a recessive allele for black body, lowercase g. Explain how you determine if the genotype of a gray fly is homozygous or heterozygous. 4.2, in fruit flies, males are XY and females XX, a cross between a grey-bodied male fly, God, I can't speak, and a black-bodied female fly produce some black-bodied females. Explain how this shows the gene for body color is not sex-linked. All right, like I said, best to pause the video and have a go. I'm going to go through the explanation now. So if you wanted to have a go and you don't want to see the answer, then pause now. All right. Here we go. Right, let's look at this first one. So we've got a fly, and it could be big G, or big G, little g, because big G, little g,
is grey, and big G is grey. Hey, why is grey spelled Grey? I don't know. Anyway, yeah, I thought it was Gray. Maybe not. Okay, right. Anyway, I'm going to move on. Lowercase gg is black. So if I've got a grey one, how would I know whether it was big G, little g, or big G, big G? Now, you can't tell from looking at the fly. You're going to have to breed it, going to have to get it on with a black fly, the grey fly and a black fly, get them to reproduce, and then you can look at the offspring. Now, the black fly is going to be little g, and so if it's homozygous, the grey fly for big G, then all of the offspring will be grey because they're all going to receive a big G from the grey fly. So I'm showing you the cross here, monohybrid cross. It's called monohybrid because we're just looking at one gene with two alleles. And that's a monohybrid cross. So you can see there all of the offspring would be heterozygous, big G, little g. They'd all be grey. So that would tell you that they were homozygous dominant. Now, if you breed the grey fly with a black fly and some of the offspring are black, that tells you that the grey fly is heterozygous, must have a big G and a little g, because you're going to do a different monohybrid cross. Let's do it down here. You're going to have big G, little g, and that's going to go against little g for the grey. For the black, sorry, the black fly is little g, and that means you're going to get a couple of black offspring, right? It's going to be 50% of the offspring are going to be black flies, and the others are going to be heterozygous grey flies. Okay, that's going to do that. Explain. Oh my gosh, look how I spelt dominant. What my teacher would have done in school is put SP, done the correct spelling, and circled the spelling mistake. I don't know if they do that anymore. They probably don't. Anywho, right. Let's go to 4.2. In fruit flies, males are XY and females are XX. A cross between a grey-bodied male fly and a black-bodied female fly produce black-bodied females. Some black-bodied females. Explain how this shows the gene for body colour is not sex-linked. Right, well, we know we've got a grey-bodied male fly. And what else? Mentioning about sex linked, so we know it could be XG, XGY. So if it was sex linked, I'm going to do a cross and show you. If it was sex linked, then the daddy, hold on a minute, then the male grey bodied fly, if it's sex linked, they won't have the gene on the Y chromosome. It can only be carried on the X. So they would be XG and then Y with nothing. And the female is black. So she's GG, lowercase g, because we know that the black color is recessive. Now, the flies they crossed had black-bodied female offspring. But check out when we do this cross, the gender and color of the offspring is important. If it is sex-linked, the gene for fly color, that means it's found only on the X chromosome. And this means that when the daddy has his kids with the black female, she's going to be big G, big G, little g. And so going to have two grey females. It's impossible to have black females in this situation. So a grey-bodied male fly with a black female fly, if the gene is sex-linked, cannot have any black offspring. The only any black female offspring. They could have some black offspring, but they'd have to be male, and I'll show you why. Because you get the lowercase g with the Y that doesn't carry a chromosome. So you would be able to get black males, but you wouldn't be able to get you wouldn't be able to get any black females. Cool. Okay, so if it was sex-linked, the female offspring would receive the grey-bodied dominant allele and so all be grey. And they'd only get one allele. The females would have to get the dominant allele from the father. Right, let's go to 4.3. A population of fruit flies contained 19% grey-bodied flies. Use the Hardy-Weinberg equation to calculate the percentage of flies heterozygous for gene G. Show you're working. So the first thing you have to memorize with the Hardy-Weinberg are the two equations. You want to do P plus Q equals 1. And then you want to write P squared plus 2PQ plus Q squared equals 1 as well. And you want to pause the video now and have a go at calculating this. All right, I'm going through the answers. So if you wanted to pause, you need to do it. So we've been told the phenotype, grey body flies, is 19%. Now we know the genotype for grey body flies is big G, big G, big G, little g. Now, when you look at this second equation, the p squared represents the homozygous dominant. It's up to you what one you give it, but I always make it homozygous dominant. And the PQ represents the heterozygous. So we know that is 0.19 as a decimal, which means we know that GG, homozygous recessive, is 0.18. If I square root that, I'm going to find out what Q is. So if I square root that, I'm going to get 0.9 for Q. I'm going to get P plus 0.9 equals 1. So I'm going to do 1 minus 0.9, and that's going to give me 0.1. So now I know that P is 0.1. I can do 2PQ. I can do 2 times 0.1 times 0.9, and that is going to give me 2PQ, which is the heterozygous flies in the population, which is 0.18. Don't forget, which I often do, I circled it up here in pink, calculate the percentage. If you just wrote 0.18, you'll only get 1 mark. We need to times by 100 to put back to a percentage. Now, you can get to that 0.18 another way, but there's many ways to do it. Obviously, once you've worked out that P is 0.1, you could square that, and then you get 0.01, and you could do 0.19 minus, you could do P squared minus 0.01, and that is going to give you 0.18. What am I waffling about? Anyway, 4.4. In fruit flies, a gene for wing shape has a dominant allele for curly wings R and a recessive allele for normal wings R, lowercase r. The alleles for this gene are on a different pair of chromosomes from the gene for body color. So what we're being told is that the curly wings and the body color are on two different chromosomes. That means they're not going to be, there's going to be no autosomal linkage here. I'm guessing that's what they're trying to tell us. Fruit flies that are homozygous dominant for curly wings do not survive beyond the embryo stage. So that tells us that if we're big R, which is curly wings, we're dead. We don't make it beyond an embryo. Okay. Curly winged fly is homozygous for grey body color. So that's big G if you're homozygous. With it's a curly winged fly. So it's got to be big R, little r. Why can't it be big R, big R? If it was big R, it'd be dead. So it wouldn't live past the embryo, so you can't breed with it because it's going to be dead. It doesn't grow to be a baby or any stage of a fly's life. Then we have a curly winged black body fly. So we know the curly winged is the capital R is dominant, but it mustn't be homozygous, otherwise it's dead. So big R, little r for the curly winged. And the black body fly is going to be lowercase g, lowercase g. It's going to be homozygous recessive for the body color. Complete the genetic diagram to show all the possible genotypes and the ratio of the phenotypes expected to develop into adults from this cross. Why don't you pause the video and have a go? All right, the first mark is for the genotypes of the parents. So I've just done those up here. So we have big R, big G, big R, little r, big G, and then we've got big R, little r again for the curly wings and little g, little g. Now I'm going to have to do a dihybrid cross here, and these ones are a little bit long. You're going to make, they're worth knowing. Now, there's probably a shorthand way to do this, and most people can probably do this in their head without doing that. I'm not one of those people. I have to do it the long way, and it might be that I have to make up time on other parts of my exam because I'm going to waste 5 minutes on 2 marks here. So we've already scored a mark, and I'm going to waste 5 minutes on another 2 marks, which is a bit of a pain in the ass, but it's the only way I know to do it. So what do I want to do? I want to work out what gametes these parents are going to make. So remember, when the male needs to make its sperm, it has to put an R and a G into the gametes, one of each allele. So it could be big R and big G, and another sperm could be big R, big G, and then we're going to do little r, big G, and little r, big G. So that is all the possible gametes for that parent. Now, the possible eggs or sperm for this parent would be big R, little g, big R, little g, are the possibilities. And we've got another big R and a little g. And then they could do little r, little g, and little r, little g. And they're the 4 possibilities for the gametes they can form with these 2. Now you just got to smash them all together. So I'm just putting all of these together just by crossing them. Nice and simple. Always put the big letter in front of the small letter when you're combining the alleles. So put the R's together and the G's together, and put the big letters in front of the smalls, or the dominants in front of the recessives. And then what we're going to do from that is work out the possible phenotypes. There's not as many as you think. Obviously, when you're doing dihybrid inheritance, you're going to get 16 in the offspring normally, but we've got a little exception here because we were told anything with a big R is dead. So we might want to take them out of the situation. I'm typing now because I was getting sick of my crappy handwriting. Right, these four are dead up here, aren't they? Those four have got big R, big R. So my possible genotypes in the offspring are big R, big G, little g, and I've got little r, big G, little g. And it's in a ratio of like 2 to 1. So um, of the big R, big G, little g to little r, big G, little g. So yeah, these are all dead, so we don't want to really include them in the ratio there. And then these ones are dead, so they don't make it past embryo stage. So are they really offspring? I don't know. Oh my gosh, I can't find the oh, there it is. Right, let's check out the phenotypes. So the phenotypes, well, we've only got two really, because the big R, they don't make it. So curly grey, which are homozygous for the curly, don't make it. So curly, we've got curly grey, big R, big G, little g, and we've got normal grey, which is little r, big G, little g. So they're all grey, but we've got some curly and normal. Now we can count these up, and it's going to be 8 of those, and there are 4. I've circled them here. There are 4 of the normal grey, and then we can simplify that to 2 to 1, and there you go. Hope you found that useful. And if you did, don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you lot on the next vid.
Hey, level biology, you are the only one I want. I me, you are the only one for me.
Yeah, if you want that A-star in A-level biology, you got to get to love it. All right, people, welcome to Let's Get to the Marks. I'm going through an A-level biology walkthrough question on the use of quadrats for sampling. A weed is a plant growing where it's not wanted. Fat henweed, which grows in some crop fields. Fat hen is a weed which grows in some crop fields. Describe it to estimate the number of fat hen plants in a field. All right, guys, hit the pause on the video. Let's go. Okay, so what are we going to do? Well, first things first, what you do if you want to sample a field, why do we sample? You might be asked why we sample, and that's not the question here. But if you're asked why we sample, it's to save time. I mean, there might be thousands of henweeds growing, or whatever they're called, fat hen plants growing in that field. You can't possibly have the time to count all of them. So turn the field into a grid, map out the field, might be a better way to write it, and then draw a grid on the map. Use a random number random number generator to generate numbers randomly. You want 10 different numbers generated on your calculator. Place the quadrat on that specific place on your grid, on the field. Count the number of fat hen plants in the quadrat. Now do this 10 times. So you might find on average there are 7 fat hen plants, because you're going to add it all up. So if it equals 70, you divide by 10, you got 7 is your mean number of fat hen plants in a quadrat. So you place your little quadrat, your little wire quadrat with little squares, into your grid position based on your random number you generated. So you're going to get a mark for saying draw a map of the field, make a grid, randomly generate 10 numbers, repeat 10 times, calculate the mean number of fat hen plants per quadrat. So that might be 7, it could be 10. It's up to you. Now calculate the size of the field. So you're going to do, the width times length, etc. It's unlikely that it's going to be a beautiful square like this, but it might be. So 40 meters times 30 meters in my example, just so that you can visualize it, is going to be 1,200 meters or 1,200 meters. Now divide the area of the field by the area of the quadrat. Now it's really handy if you have a 1 meter square quadrat, because then it's just, well, if it's a 1,200 meter square field, then you can fit 1,200 quadrats in there, but unlikely. Something like a 0.25 meter square quadrat is more likely. So you're going to divide 1,200 by 0.25, and then you're going to know I could fit 4,800 quadrats in my field in total. We know what the mean is, 7. So what's going to be the total number of plants? If there are on average 7 plants per quadrat and 4,800 quadrats, you're going to do 7 times 4,800, and you're going to get something like 33,000 fat hen plants in the field. So that's one way you could do it. You could also say, another way to do it is to say, okay, my quadrats are 0.25. So if the mean was 7, I'm going to go, well, there are 4 of those quadrats in a meter. So 4 sevens are 28. And I'll say, well, there'd be 28 per meter. So I do 28
times 1200, you're going to get the same answer. So you could do 28 times 1200, or you could do my way. So it's up to you which way you prefer. And that's going to get you all of the marks for this sampling quadrat question. Remember, the reason why we sample is to save time. The reason why we make it random is to make it non-biased and representative of the population. Okay, let's move on. In potato plant fields, fat hen plants can grow up to a height of 2 meters and absorb large quantities of nutrients from the soil. Fat hen has a negative effect on the growth of potato plants. Use the information provided to explain why. In your answer, name the type of competition occurring between fat hen plants and potato plants. Okay, so fat hen plants and potato plants, we can assume are different species. So what type of competition do you get between two different species? You get a certain type of competition inside a species and a certain type of competition between two different species. So you can see my fat hen plant here blocking out some sunlight to my potato plant. It's also going to affect the nutrients that are absorbed through active transport into those root hair cells in the soil. What you need to do is explain how the presence of that fat hen plant is going to negatively have an effect on the potato plant. So pause the video, have a go. All right, the competition is called interspecies competition. What else? We can say sunlight is blocked by the height of the fat hen plants because they say they're 2 meters tall. Means less light is absorbed or is available for the potato plants, so they can do less photosynthesis, produce less glucose for energy for growth. We could also say a less nitrates absorbed, through the root hair cells because the fat hen plant is sucking up all the nitrates. And if you have less nitrates, you can't react them with glucose to make amino acids. And if you don't have amino acids, you can't polymerize them into peptides and proteins are needed for growth. You get the idea. So you just want to go through the steps, put the key points down and smash out all of the marks. Okay, maybe I get a little bit excited when I'm doing these walkthroughs, get a bit hyper, but that's probably due to my caffeine addiction. I tend to drink about 12 cups of coffee a day while I'm teaching online and doing walkthroughs and working from home, tutoring and teaching. I just drink far too much coffee. I never had the opportunity to drink that much coffee when I was a teacher in school, but I take full advantage now. Right, 5.3. Table 1 gives some features of fat hen seeds. Mean mass in milligrams is 0.77. Maximum number produced per fat hen plant during a growing season is 20,000. Percentage viable able to develop after a growing season is 79. During a growing season, it was estimated that a total number of 550 fat hen plants grew on the fields of a farm. Calculate the maximum mass in kilograms of viable fat hen seeds on this farm after a growing season. Show your working. Assume that all seeds produced during the growing season remain on the farm. It's worth 2 marks. Grab your calculator, pause the video, have a go at this one, then unpause and watch me go through the explanation with the calculation. Let's go. Okay, so we can see the mean mass is in milligrams here, 0.77, but we actually need to convert to kg. Look, I circled it down there in the answers. So you've got to know your steps. When you're moving down from grams to milligrams to micrograms to nanograms, you multiply by 1,000. So it's kind of counterintuitive. As you go smaller, you times by 1,000. As you go bigger, you divide by 1,000. So I'm going up from milligrams to grams, divide by 1,000, and then from grams to kilograms, divide by 1,000 again, or just say divide by a million, 1 times 10 to the 6. So I want to go 0.77 milligrams divided by 1 times 10 to the 6 divided by a million means you have 7.7 times 10 to the minus 7 kilograms. That's how much each seed I was going to say weighs, but that's the mass of each seed. Now, how many seeds does a plant make in a growing season? Well, it makes 20,000. So one plant makes a seed with a mass of 7.7 times 10 to the minus 7 kilograms, but there's 20,000 of them. So I need to times by 20,000 to work out the mass of all the seeds made by one plant. And if you times that by 20,000, you're going to get 0.0154 kilograms. Which is the mass of all the seeds that one plant can produce. However, we have 550 plants on the farm, so we're going to times that by 550. And if we times that by 550, you're going to get something like 8.47 kilograms. Now, we've got another factor here. Only 79% are able to develop, are viable. And our question specifically asks us, calculate
calculate the amount of viable fat hen seeds, the mass of that. So I want to do 79% of 8.47 kilos. So I'm going to do 79 divided by 100, 0.79 times 8.47. I'm going to get 6.6912, something or other. So I'm going to make that to 3 sig fig, 6.69. And there we go. You're going to get both the marks. Don't forget Marty Tucker's sharing's caring. Hit the like, punch up the bell, subscribe. I'll catch you lot on the next video. Let's smash them A-level biology exams. Come on. Welcome to Let's Get to the Marks. Going through a quick walkthrough question on the myogenic stimulation of the heart and how to use p-values. Okay, guys. Let's get into it. 6.1, describe the myogenic stimulation of the heart and how the regular contraction of the atria and ventricles is coordinated. Do not include the autonomic nervous system in your answer, so no vagus, sympathetic or parasympathetic nerve and acetylcholine. You don't need to talk about that business. Just about the myogenic stimulation of the heart. Why don't you pause the video and have a go at this one on paper with a pen, and then I'll go through the answer with the explanation. Let's go. Press pause. Okay, so I'm going to sketch out the heart here for you, man and girl there, just to help with the old explanation. I suggest you learn how to do these heart drawings off by heart. I got a level 4 in GCSE art, so if I can learn how to do it, you can too. It helps you with actually visualizing the heart when you get questions on the heart. If you can learn to scrap it out on paper, and you can do that while you're watching Netflix or Prime or whatever you want. Yeah, you're going to make a few mistakes like I have here, but you're more or less going to get the hang of doing it. And once you've done that, you pretty much got the heart locked down, the structure of it. Not that you get a lot of questions on that, but it will help you answer other questions where you've got to apply your knowledge. Now, our myogenic stimulation of the heart starts with the right atria, and inside the right atria is the S.A.N, the sinoatrial or sinoatrial node. And this node sends out an electrical wave of depolarization. You can call it an impulse, but it's going to send out this electrical wave across the atria, causing them to contract at roughly the same time. Now, this electrical impulse or wave of depolarization, why doesn't it cause the ventricles to retract? Surely it can go downwards. Well, we have a band of what we call non-conducting tissue that is almost all the way across the bottom of the atria, blocking the electrical impulse being able to be conducted down into the ventricle muscular walls. So we're just going to get the atria to contract, and this is going to provide us with a little delay between the contraction of the atria and the contraction of the ventricles. So why do we need that delay? Well, that delay is needed so that the atria and ventricles don't contract at the same time, because they're not going to empty fully there, and we're going to get some backflow of blood if that happens. So we do need that delay. Now, there is a conducting tissue down the septum, which is the middle of the heart, and this is called the bundle of His, and there's also fibers that reach around from the bundle of His and work their way up from the apex, the bottom of the heart, upwards through the muscular walls of the ventricles, and these are called Purkinje fibers, and they will conduct the electrical wave up from there. So let's put this down in can't speak. Let's construct our answer. The SAN, the sinoatrial node, releases a wave of depolarization, causing the two atria, the right and the left, to contract simultaneously. However, we want to say the non-conducting tissue prevents this wave of depolarization reaching the ventricles. So there is a gap or a delay, time delay, between the atrial contraction and ventricular contraction. Of course, this is going to ensure that these completely empty and we don't get the backflow of blood. It makes for a much more efficient system. Now there is a second node, and depending on the atrioventricular node or atrioventricular node, put in the comments which one it is, I can't remember. But the second node is situated, as the name says, as it says on the tin, AVN. It's between the atria and the ventricles. Sometimes you'll see that it appears in the kind of atria side, and sometimes they show it on the ventricular side. So it kind of crosses both. Now, once that is stimulated, it will also release a wave of depolarization or an electrical impulse that can only go down through the septum. That's the center of the heart, the wall that divides the two ventricles, can travel down a tissue through there called the
bundle of His, a bundle of special nerve fibers. These break out into Purkinje fibers, other nerve fibers that spread out throughout the muscles of the ventricular walls, delivering the ventricular wall contraction from the bottom upwards. Now, you don't have to know septum on your syllabus or apex, but they're good words to know to describe what's happening. So rather than saying the ventricles contract from the bottom upwards to ensure they fully empty, we can say they contract from the apex upwards. So it's just a good descriptive word to help you explain what's going on. We call that the apex at the bottom there. Okay, and that really is going to get you all the marks. I know I put 6 ticks, but my answer's worth 6, not 5 AQA. So hush. Right, we're going to move on to the next part of the question. So let's go. 6.2. Atrial fibrillation, AF, is a condition that causes an irregular heart rate. Scientists used a statistical test to investigate the association between different factors and the risk of developing AF. By the way, when they put AF in brackets and they give you an abbreviation, you can use it in your answer. You don't have to write atrial fibrillation. So table two shows some of the scientists' results, including probability values P obtained using the statistical test. We've got age, high blood pressure, high LDL concentration, that's low density lipoprotein, and hyperthyroidism. Look, there's my complete healthy wave, and then atrial fibrillation is where your wave is all irregular heartbeat all over the place. Right, we've got these probability values. So for age, it's 0.004. For high blood pressure, it's 0.001. For high LDL, low density lipoprotein concentration in your blood, it's 0.222. And for hyperthyroidism, it's 0.018. What can you conclude from table 2? It's worth 3 marks. Why don't you pause the video and have a go? Guys, at this point, I don't have to stress, but it will massively help me out if you guys can hit the like. It really, really helps me out. Okay, let me go through how you'd go through this. Okay, so p-values. What you got to know about p-values generally is that at A level and in most biological situations, a p-value less than 0.05 is significant and means that an association is significant between the two variables. Now, if it's greater than 0.05, it means it's not significant. So we can say here that age being 0.004, lower than 0.05, high blood pressure extremely significant, 0.001, and hyperthyroidism is significant but not hugely, 0.018. So it's still lower than 0.05. So I want to say age, high blood pressure, and hypothyroidism are all show a significant association with AF, atrial fibrillation. We can reject the null hypothesis in all the cases for this. The null hypothesis is obviously the one which says there is no significant association between these factors and AF. So, however, I also might want to put some nuance here and say that high blood pressure is highly significant. And I could say that hyperthyroidism is low significance, but still significance. Now, so this means that it's unlikely that these are associated down to chance. I don't know what that is for high blood pressure, 1 in 1,000 or 1 in 10,000, whatever it is. But the chance of it being down to just random chance, the association is very small. Now, if we look at the high LDL, low density lipoprotein concentration, it is above 0.05. This means that it's greater than 0.05. So any association is likely down to chance. And we would say that this is not a significant association between high LDL and atrial fibrillation. And that's going to get us all at a mark. So just work out how to do these p-value questions and you will be sorted. Okay, now I might want to say for high LDL, we would accept the null hypothesis and say there is no association between high LDL and AF. Booyah! Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. I'll catch you lot on the next video. Let's smash them exams together. Let's go. Welcome to Let's Get to the Marks, going through an A-level biology biotech question on genetic engineering. Let's get into it. 7.1 BAM-H1 and HIND3 are both restriction endonucleases. Figure 4 shows the positions where these enzymes cut a linear, means a straight chain, molecule of DNA. We got position DNA molecule cut by BAM-H1 shown by a line, so just a straight line. So you can see it's cut into 3 pieces, that DNA molecule, if you just had those lines on that rectangle. Now, the arrows show the position of the DNA molecule cut by the HIND3 restriction endonuclease.
So they've cut it in two places, both different enzymes. Two experiments, multiple copies of the DNA molecule shown in figure 4 were all completely cut into fragments using these restriction enzymes. The DNA fragments produced were separated by electrophoresis. Experiment 1 cut the DNA fragments using the BAM-H1, that's the straight line. Experiment 2 did the line and the arrow. Complete figure 5 to show the relative positions of the bands following electrophoresis. We got this empty square here with experiment 1, BAM1, and experiment 2, BAM1, HIND3, and the direction of electrophoresis on the right. Now, in order to answer this question, we have to complete that figure. So I'm going to draw it here. Okay, I'm going to zip it out here and just do a copy down there because I can't be flipping the screen up and down too much. So if you haven't seen one of these electrophoresis diagrams before, what you've got to do is you've got to draw lines on where you think the different fragments would move. Remember, DNA is negatively charged, so it's going to move from a negative electrode to a positive electrode through the gel. And the smaller the fragment, the further it's going to move. So complete figure 5 to show the relative positions of the fragments with each two experiments. So experiment 1 are circled here in pink using BAM-H1. By the way, pause the video, see if you can draw the pattern, and then unpause and watch me go through it. Okay, let me just zing it out, actually. Let me quickly draw it out. Let's delete that. All right. We're looking at experiment 1 now. So hopefully you pause the video, had to go on some scrap paper. And you can see if we just look at experiment 1, there was no HIND3, so we don't use the arrows. We're going to get a small fragment, a slightly larger fragment, and then a really big fragment, right? It's cut into 3, small, medium, large, whatever you want to say. So we need to represent that on experiment 1. Now, the large fragment is going to move really slowly. But when you finish the electrophoresis, it hasn't moved very far. That's that first one. And the two smaller ones you can see have moved down here. Okay, so we've got 3 lines. But now we're going to look at Experimente 2. Now, basically, the diagram they've given us in the exam paper is experiment 2. So we got this smaller fragment here, right? It's even smaller than the one from experiment 1. So actually, hold on, I want to do it down here. It's going to have traveled the furthest. Oh, there's an even smaller one here I've shaded in pink, and these two in pink are even smaller than our first one. So I'm going to want 3 lines down here, and they've all gone further than the smallest one in experiment 1, right? Because they're all smaller. Now check out this next fragment. This is exactly the same as experiment 1 because the HIND3 did not cut it in half. It was the BAM1. So I've drawn those on the same place. So that's going to be really important that experiment 1 and 2 have a line in the same relative position to represent that fragment. Now look at the largest fragment in experiment 2. It's not as big as the largest fragment in experiment 1 because it's got that little bit cut off by the BAM1 to make that smaller fragment. So the largest line is not going to be as far back as the BAM-H1 line because it hasn't traveled as far. Hopefully that all makes sense, peeps. Leave a comment, by the way, if you think I'm going through the videos too fast, you'd like me to slow down and talk through them a bit more slowly because I do have a tendency, I have a tendency to waffle, and I have a tendency to go too quick sometimes. So I want to get the right speed, whether it's better for me to just go slow and talk it through slowly or what. Anywho, I'm going to drag this image over here because we're going to use this for this next question. So let me just bust out the pattern here because you'd have drawn this now in your exam. We'll assume you got this right. Let's get rid of all these other bits of the drawing. We don't need the negative and positive. We never needed that. And we don't need that arrow. And we don't need the ticks, really, do we? Nah, we'll get rid of those ticks. Okay, so we got our pattern that we've drawn on and we've scored both our marks. We're going to look at 7.2 now. So just how you could determine the size of the different DNA fragments produced in these experiments, and it's worth 2 marks. Okay, so how would you find out how many kilobase pairs are in each of these fragments? So what you're going to do, you basically are going to run it's a bit like doing a calibration curve when you do the Benedict's test. When using colorimetry, you produce a calibration curve. You're going to run known sized fragments. So say you had 20 different fragments of DNA and you know the size, 10 kilobase pairs, 20 kilobase pairs, etc. You would run those through the gel for the same amount of time, pause it, take your print and look at the position and write that down, and you make a kind of ladder, a known ladder that you put next to the gel, and then you can run your unknown, i.e. what we've got here, and you can just read it off by comparing with your known DNA fragments on the DNA ladder and your unknown, which will be in the gel. So you create something called a DNA ladder, or a ladder of fragments of known sizes, and that's going to get you the marks. So let me just draw that on there. So now you can see in pink here along the right, I've got different fragments of known sizes, and then you just read off your fragment. Like my BAM1 first fragment would be about 100 kilobase pairs. I read it off against the ladder of known ones that I've put on the right hand side. You get that idea, right? And then you look at the next one, maybe that's 110 or whatever. This one's like 400, 410, whatever. So you just say compare your unknown DNA fragments to your known DNA fragments on the ladder. Right, 7.3, how many DNA fragments would be produced in experiment 2 if the original DNA molecule was a plasmid? So we're going to go back and then describe how the restriction endonuclease, the DNA ligase, are used to insert a gene into a plasmid. So how do you use those two enzymes to insert a gene? Pause the video, have a go. Well, I'm going to go through the answers. You might have already had a go at this one. You can see this is really from experiment 2. There are 5 fragments when you used all the enzymes. But what about if we join the ends up? Imagine you take that long rectangle, you curl it round and stick the ends together. How many fragments would you have then? So what you're going to do, just to sort of try to explain it a little bit more clearly, let's just zing that out. You're going to have like fragment 2 here. You can see it the same as on the rectangle, then 3 there between those two enzymes. And then between these two cuts with the hind 3 and the BAM H1, you've got 4. And then you've got the big 5. But look, the 5 and the 1 would actually join up to make just one fragment. When you join those ends together, the 5 and the 1 have no cuts between them. So they would become one fragment. I don't know what that is. So then actually, that looks like an F rather than, I think I drew the 4 sideways. Looks a bit weird. All right. There you go. 4 fragments. If you join the ends up like that, bish bosh, there'd be 4 fragments, not 5. Cool. Okay. Let's move on to 7.4. Now, I recommend pausing the video and having a go. You just got to explain how these two enzymes, it's only 2 marks, are used to insert a gene in a plasmid. So pause and have a go. All right, what you're going to do, you're going to cut the plasmid with the restriction endonuclease, and that will create what we call sticky ends. As you can see, I've attempted to draw them on my plasmid there like a jigsaw piece. And then you're going to use the DNA ligase to stick the fragment into the plasmid. And it does this by forming phosphodiester bonds. And that's basically going to get you all of the marks. So, yeah. Don't forget, sharing's caring. Hit the like, punch up the bell, subscribe, read through it, have a go on some scrap paper, see what you can do, and then unpause and watch the explanation. All right, let's go. Plant cells are surrounded by a rigid cell wall. Cell wall plasticity refers to the ability of a cell wall to stretch permanently. IAA is a growth factor which can activate enzymes that loosen the cell wall. Scientists investigate the effect of IAA concentration on cell wall plasticity and the growth of stem segments. Here's my cell getting its wall a bit loose and doing a bit of cell elongation. Okay, figure 6 shows some of the scientists' results. There's figure 6. You can see we've got cell wall plasticity in arbitrary units on the left hand side with the dashed line, and we've got the mean growth of stem cells in millimeters with the solid black line. So it's one of these biology graphs where you have 2 y-axes. Make sure you only read cell plasticity from the left and mean growth of stem from the right hand side. And then at the bottom on the x-axis we have the IAA concentration, 10 to the minus 8 all the way up to 10 to the minus 5. So it's increasing the concentration as we go from left to right. Use the information provided to explain the relationship between the IAA concentration of cell wall plasticity and the mean growth of stem cell segments. It's worth 2 marks. Why don't you pause and have a go? And the keys here is to use your pen to highlight keywords in the question, in the stem of the question. So use your science content. That's what the word explain means. So you can't just describe the graph. You've got to use your science content. Now it's 2 marks. You're going to get 1 mark for a description within the explanation and then 1 mark for the scientific content. Okay, so we need to comment on the relationship between IAA and the dashed line and the solid line, the cell plasticity and the mean growth of the stem. So I'm just going to say, as we increase the IAA concentration, or as IAA concentration increases, so does cell wall plasticity, and we can say, and the mean growth of stem segments. Okay, now I put a little question mark here. We need an explanation. So why does the cell plasticity increase? Well, if we look up in the stem, they told us that the IAA is a growth factor which activates enzymes that loosen the cell wall. So I'm going to put that in my answer. This is because it activates, the increase in IAA activates enzymes that loosen the cell wall, and therefore the cell elongates. And this would explain why we get more growth of the stem. And that's going to get me all the marks. I know I might be over explaining it here, but this enables cell elongation to occur and the mean growth of the stem to increase. So I've kind of pointed out, I probably should have said as IEA concentration increases, so does cell wall plasticity and mean growth of stem. This is because it activates enzymes that loosen the cell wall and enables cell elongation. But I included the increasing stem at the end. Okay, 8.2, use figure 0.6. Use figure 6, not 0.6, to calculate the percentage increase in the mean growth of stem segments from the IAA concentration when it was increased from 10 to the minus 8 to 10 to the minus 5. Give your answer to two significant figures. It's worth 2 marks. We're going to need to go up and look at the graph for this one because we're going to have to use the graph. So let's go up and calculate the increase in the growth of the stem, not the plasticity of the cell wall. So we're going to calculate the percentage increase in the mean growth of the stem. That's the solid black line. And we're going to use that information. And it's between minus 10.8 and minus 10.5. Why don't you pause the video, bust out your calculator, have a go at this one. Okay, folks, so what we want to do is at 10.8, we're looking at 0.6. Don't forget to look to the right-hand side, not the left-hand side. All right, that's super important. And then when it is at 10.5, the IAA is increased. You can see the mean growth goes up to 4 millimeters. So at 10.8, it is 0.6. We're using the right-hand side for the mean growth of stem. We're not looking at the left axes there, the y-axis on the left. And so we're going to do the difference, and it's 4 minus 0.6. The difference is 3.4. Don't forget, with doing any percentage increase or change, you need to remember that you're looking at the original over the difference over the original, or the change over the original times 100. So the change is 3.4 because we go 4 minus 0.6. The original was 0.6, was the original growth of the stem, and then we're timesing 100. So 3.4 over 0.6 times 100. And don't forget, we need to do this one to 2 significant figures. Gosh, I'm all over the place today, folks. Okay, let's go. Okay, and my final answer is 566.66%, but they said 2 significant figures, 2 SF. So we're going to go, the 6 is bigger than 4, right? So even if it was 565, we would round up to 570. So even if it was 565, you're going up to 570. You don't count the 0 in the significant figures, but you have to put it there. All right, gibberellic acid GA is another plant growth factor. A student was asked to design and carry out an investigation into the effect of different concentrations of GA on the growth of stem segments. The student was provided with 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5 mole per decimeter concentrations of GA solution, distilled water, 6 petri dishes, and access to glassware. 60 stem segments of different lengths. So they've been given 60 stem segments of different lengths. A technician produced the different concentrations of GA solution from a stock 10 to the minus 1 concentration of GA. Describe how the technician produced 10 to the minus 3 mole per decimeter solution. Right. Why don't you pause the video, have a go at this one, and then I'll go through with an explanation. Okay, so if you started off with a 10 to the minus 1 mole per decimeter concentration of GA, how could you dilute it down and produce a 10 to the minus 3 solution? Well, first of all, you start with 10 to the minus 1. And if you want to go to 10 to the minus 2, that's 0.01, and 10 to the minus 3 is 0.001. You're just diluting by a factor of 10 each time. So you start off with your 10 to the minus 1 stock solution, right? It's 0.1 mole per decimeter. I want to take 1 centimeter out of that and add it to 9 centimeters of distilled water. That has diluted it 10 times. Now, I know people think, hold on a minute, isn't the ratio 1 to 9? When you're looking at the amount, you've got to think the total volume is 10. 1 of the stock, 9 of the water. So it's a total volume of 10. You have 1 centimeter of 0.1 in a total volume of 10. So you've diluted it 10 times. Now you repeat that again using the new 10 to the minus 2 as your stock. So you take 1 centimeter of the 10 to the minus 2, add 9 centimeters of distilled water, and the total volume of 10 to the minus 2 is 1 in 10. So it's diluted 10 times again to 10 to the minus 3. So we're going to put that into words. Take 1 centimeter cubed of stock, add it to 9 centimeter cubed of distilled water. This makes 10 to the minus 2 mole per decimeter, 10 centimeters of it. Now take 1 centimeter of your 10 to the minus 2 mole per decimeter and mix with 9 centimeter cubed of distilled water. You now have 10 centimeter cubed of 0.001 mole per decimeter, or 10 to the minus 3. Now you could do, it doesn't matter with the volumes. I just did 1 and 9. You could use any amounts. Okay, you could use any amounts. So as long as it was 1 to 10, 1 to 9 in your parts. Right, describe a suitable procedure the student should follow to investigate the effect of different concentrations of GA on the growth of stem segments. In your answer, you should include the variables that should be controlled in this investigation. It's worth 6 marks. Pause the video, have a go on paper, see how many bullet points you can get down that make sense. All right, this is the way I'm going to do it, right? We're going to take 5 different concentrations of GA. So we're going to go 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. You don't need to put the concentrations, just tell them you're doing 5, right? Do at least 5 different ones. We're also going to use a control. So we're going to do distilled water, and they've given us 60, right? They're giving us 60 plant segments. So how many can I put in each? Bearing in mind, I've got 5 concentrations and 1 control. I can put 10 plant segments in each dish, Petri dish or whatever dish you're using. So I've got my control, which is just distilled water. I'm going to add 10 stem segments to each of the dishes. So 10 to that one and 10 to each of my concentrations. Remember, before we add them, we need to measure their length because we want to see the effect on growth this GA is having. So we're going to measure the length beforehand with a ruler. You could use some waffle about how you're going to do that if you really want to. It's not going to get you extra marks. And then you're going to leave for a set time period, maybe 24 hours, 48 hours, 10 hours, whatever, 2 days, 1 week. Just make sure it's the same time period for all of them. Then measure the final length once you take them out and dry them off. Measure the final length and calculate the difference between the starting and the final length, your change in length, and then do that as a percentage. You could write down how to do that, but you already did that in an earlier question, so you don't need to worry about that. And then you could say plot a graph of the mean segment length against concentration if you really want, but I don't think that will get you extra marks, but it's just something that you might want to talk about because you're thinking your way through it as you go. Now, we did do 10 segments in each, so we should find the mean change in length at each concentration. And I probably should have said calculate the mean change in length at each concentration. And I could have said remove any anomalies. So there'd be an anomalous one if one of them has shrunk. Imagine one of them has shrunk and the other 9 segments have increased. So we might say that's an anomaly and then remove that from our calculation. Now, they've specifically said, talk about the variables that should be controlled. So you want to list about 3 or 4 of these variables. You could say how you're going to control them, but they didn't say that. But I'd put it there just in case. So I could control the temperature using a water bath. You could place all these dishes in a water bath. You could measure the diameter, make sure they've got the same thickness or same diameter at the start. You could have the same initial starting length of the stem, so you cut them all to like 20 millimeters or whatever you want to start it at. Same age of the plant, so maybe you're using a 6-month-old plant. If the plant was too old, it might not grow as rapidly, might not have as many meristematic tissue. Leave for the same time period, and that should cover all of the stuff and get you all of the marks. All right, guys, don't forget sharing's caring. Hit the like, punch up the bell, subscribe. I'll catch you lot on the next video. Do me a big favor, hit the like, guys. Welcome to Let's Get to the Marks, going for an A-level evaluation question of an investigation in A-level biology. Let's get into it. So question 9, a person with diabetes is in diabetes remission if their blood glucose concentration is below the diabetes threshold concentration for at least 3 months. This diabetes remission is achieved without taking medication. Scientists investigated whether a weight loss program would result in type 2 diabetes remission. The scientists used a computer-generated list to select 380 volunteers from a large number of health centers. So
What I recommend is when you're reading through something like this, obviously, if you're watching this on a screen and you haven't printed it off, which why would you, you're going to struggle with underlining and circling bits. But what I'm trying to just show here is that's what you should be doing. You're sort of reading through when you get one of these investigation evaluation questions towards the end of paper 2 for your AQA A-level biology. You're going to get these evaluation questions. And so you're immediately going to want to know what's the size of the sample, what's the age range. So they selected volunteers aged 25 to 60 years. That's a pretty good representative age range of the population. They only did it with people who had diabetes for 5 years. That might be a negative for less than 5 years. And then they evenly divided. I think that's good. The 380 volunteers were divided in a 1-to-1 ratio, so 190 in each group, into Group P and Group Q, which was a control group. They had a control. That's really good for making a comparison, so you can see if your independent variable actually affected the data. And then they placed Group P on a weight loss program for a 2-year duration. That's very long term. So that's another really good thing about this study. It's going to make it reliable. They recorded the loss of mass and percentage of volunteers in each group in type 2 diabetes remission after 2 years. Now, the first part of the question, 9.1, give 2 reasons why a weight loss program could be used to treat diabetes type 2, but not diabetes type 1. So basically asking you, do you know the difference between diabetes type 1 and diabetes type 2? So it's just basically what is the difference, and that is going to get you some marks. So diabetes type 1 is where you don't produce insulin at all, and therefore, and weight loss or weight gain is not linked to affecting diabetes type 1. It's actually about you don't produce any insulin. So that's what diabetes type 1 is. So losing weight is not going to change that. Diabetes type 2 is linked to weight gain. So obesity and stuff heavily correlated with the development of diabetes type 2. Now, diabetes type 2 is different. You still make insulin. So here's my healthy cell with my insulin with the circled in pink with the eye and the receptor on a liver or muscle cell. The difference is once you've got diabetes type 2 with the drawing down below, they're no longer complementary, the receptor and the insulin. It's not the insulin that's changed shape. It's the receptors that will no longer bind correctly with the insulin. You won't get that cascade effect or whatever effect that goes on within the cell. So, yeah, so you're just pointing out your difference between diabetes type 1 and type 2 there. 9.2, the design of this investigation helps to support the validity of any conclusions obtained. So, what is it about this that makes this valid? What are 3 things that you would say about this experiment that makes it valid? You can pause the video, have a read through and see, but you can see there's loads of good things they've done here. They use computer generated lists to select 380 volunteers. So it's a large population, a large sample size, sorry. So that is really good. They did the computer generated selection, which you always should have a random selection, and that avoids bias. So you need to put the explanation of how it helps. So a large sample size and random. Large sample size makes it representative of the population, and random avoids bias. The fact that it came from different health centers also makes it more representative because you're going to get people from different backgrounds, different economic, basically different regions, you know, all over the country. Now, a really good age range, 25 to 60 years. That's a 35-year age range. You're going to get a nice representative spread of the adult population. They divided the volunteers in a ratio of 1-to-1 between the experimental group and the control group. So I'd say, look, they did a control as well, which is great because that enables you to make comparisons between your experimental group and the control and see if what you're changing, the weight loss program, actually has an effect compared to a control group. The fact that they did it on a 2-year duration, that means it's a long-term study. I would add the word that makes it reliable because it's been done over a long period of time. So we know that any results or conclusions that can be made are long-term ones. Yeah, and I guess that's enough, really. We only wanted 3 valid points about it. Now, 9.3, table 3 shows some of the scientists' results. So we've got some data here. You can see for Group P and Group Q. Remember, Group Q is the control group. Group P is the experimental group. Okay. Percentage of people with weight gain achieving diabetes remission. Okay, so Group Q are not on the weight loss program. Group P are. Use the information provided to calculate the difference in the number of volunteers between the 2 groups that achieve diabetes remission. So Group P, 42.1% achieved remission compared to Group Q, which was a lot less. We're going to look, the ratio was 1-to-1 and they used 380 volunteers. So let's skip back down. By the way, you can pause the video and have a go at this if you want. Of course, I forgot to say that with just scrap paper and pen, but I'm just going through the answers. So what we're going to do, you're going to work out how many people were in each group. Well, 380 total volunteers evenly divided in a ratio of 1-to-1 means 190 in each group. Let's times it to find out the number of people in the experimental group P that actually achieved remission. So you're going to do 0.421 times 190 or 42.1 over 100 times 190. You're going to get 79.99. This is actual people, so you could round that to 80 if you want. Lovely jubbly. Now we're going to look at Q. These are the people who weren't on a weight loss program. See just the general population that you're studying over that 2-year period. So we're going to do 190 times 4.7 over 100. So that achieved remission. Their diabetes started reversing. And that is 8.93 people or 9 people. So you can do 80 minus 9, or yeah, you get the idea. You can keep the decimals in there and you're going to get roughly, it's going to equal a difference of 71. So if you do that, you're going to get 71.06, but we don't have 0.06 people, so we're going to put that to 71. Again, we could have rounded those up to whole numbers, and that we'd achieve the same thing. So you're going to get some for the working out. So if they see 79.99 somewhere on your working, they know you've calculated one of the number of people. Use all the information to evaluate whether a weight loss program is an effective treatment for type 2 diabetes. So can we say that losing weight is effective? So we want to compare the results for P to Q. And when they use the word evaluation, I think plus and minus, AQA biology, you must say a couple of good things for, a couple of things against. There's always 10 negative things you can say about it. And even a good investigation like this, we can say some things that are negative. Okay, so, and you just need to make the statements. So look, it really sees here that the percentage achieving remission on the weight loss program, group P, is 42.1% compared to group Q. You could do the maths, do 42.1, take away 4.7. You could say the difference in the percentage achieving remission on the weight loss program. So yes, a much higher percentage achieving remission in group P than group Q. It should also strike you, is it significantly higher? We can't say that without doing a t-test. So that's a negative that we can come to in a minute. Right, the percentage achieving weight loss, more than 15 kilograms of weight loss, the percentage of people who achieve more than 15 kilograms of weight loss, and the link to the remission, we can see that yes, 15.8%, yeah, it's heavily linked to remission. And it seems even for both groups, it proves that weight loss is heavily linked to remission, because even the group that went on the weight loss program, if they lost weight and only 2.1% lost weight, 75% of those people who lost weight achieved diabetes remission. So it's not just about following the weight loss program, just losing weight above 15 kilos seems to be that puts your diabetes in remission. So these are all good things towards this experiment. These are good things in our evaluation. Now we want to look at some things maybe not so good. So we want to think about our methodology and things that they could have improved. So they were only looking at patients who had diabetes for less than 5 years. And so if you're going to make a conclusion, you'd need to include that in the conclusion, and they didn't include that in their conclusion. So we can't say it's valid for people who've had diabetes for 6, 7, 8 or greater lengths of time than 5 years. We don't know if weight loss is valid for that. There was no stats test to compare the experimental group P with Q and show that, right, it looks really big, the difference in the 42.1 to 4.7, but we don't know if that is a significant difference or it's down to chance. So we'd need to do a stats test, preferably a t-test for that. And this is only remission we're talking about here, which is a reversal or a reduction in your symptoms. So they're reversing it. It's not a cure. So the statement that we're evaluating is the weight loss program good for treating diabetes? Yes, for remission, but we've got no data on whether it cures the diabetes. Also, the age range, they looked at 60 to 25. We cannot say that the weight loss program is effective for people under 25 or over 60. So that's not necessarily negatives, but they're things that we need to evaluate and include in our conclusions. And that's about it. I'm sure there's some other bits. You can consult the mark scheme that you will find on the Amazing Physics and Math Tutor website for the 2024 Paper 2. But yeah, really good to practice all of these when you're preparing for your Paper 2. That's question like 8, 9, and 10. They tend to have a lot of evaluation and comprehension questions. Let's go. Let's get to the marks.
You're still here? It's over.
Okay, welcome to Let's Get to the Marks. Going through one of the comprehension questions at the end of the 2024 AQA Paper 2 A-Level Biology. Let's go read the following passage. The wall gecko is a medium-sized lizard in an isolated habitat of the southern Italy. Interesting. The wall gecko shows phenotypic diversity. Scientists investigated whether disruptive selection was leading to sympatric speciation in the wall gecko. Pale geckos live on walls and are nocturnal, active at night. Dark geckos live mainly on dark trunks of olive trees and are diurnal, active during the day. These diurnal geckos can change skin color when occupying different surfaces during the day. Comparison of the mitochondrial genes indicated that the diurnal geckos formed a distinct genetic group. This comparison also confirmed that all the geckos in the habitat were of the same species. The scientists used the mark release recapture method to estimate the size of the population of geckos in the habitat. Use the information in the passage and your own knowledge to answer the questions. The wall gecko shows phenotypic diversity. Lines 1 to 2 suggest two factors that have resulted in this phenotypic diversity. Now, what am I doing at the moment? You've got a big comprehension question. I'm going to spend 2 minutes, even before, just graffitiing down, making some notes, highlighting, circling key parts. So phenotypic diversity, what does phenotype mean? Well, it means when things look different. I've circled sympatric speciation here. What is sympatric speciation? Sympatric speciation is when you have speciation, but there is no geographical barrier. You're not separated by mountains or by a river or by a lake or a volcano or an ocean. You're actually living in roughly the same environment. So I'm just writing the definitions because I know they might come up in this question. Disruptive selection I've underlined. You see I've drawn this little graph. It looks like you can see the dashed line. I've done the hump with the dashed line. That is like your population, or average population, which doesn't favor the extremes. Disruptive selection favors the extremes, i.e. hairy and without hair, or in this case, pale and dark, the extremes of color that you could have. Then we've got mitochondrial genes, so that's going to be about genetics. We've got species. What's the definition of a species? We've got the mark release recapture method. Population size is first sample times second sample over the second, the number of marked individuals in the second sample. Anyway, let's get on with these questions, people. Why don't you pause the video, have a go at this first one. It's worth 2 marks. What 2 factors can result in the fact that you have pale and dark lizards? What factors? Okay, I'm going to go through the answers, guys. So that could occur by random mutation, okay, a change in the nucleotide base sequence, which is a random mutation. Or it could occur via environmental factors. When I go out in the sun, my skin goes darker, and that's an environmental response. But these random mutations could occur via very different methods. So you've got the random fertilization of gametes as well, could mean that we end up with this situation. So we've got random mutations, then we've got crossing over and random assortment during meiosis 1. Cool. Okay, so those are just some ways that we get variation. Okay, 10.2, the ability of diurnal geckos, these are the ones that are active at the day and night, to change skin color is advantageous. Why is it an advantage to be able to change your skin color? So the diurnals can go dark or light. They can change their skin color and live in different environments. This means they're better able to camouflage because they can change color to suit their background or their environment, a bit like a chameleon, right? And therefore, they're less likely to get eaten by predators. So you've got allowed to camouflage, less likely to be eaten by predators. You could go that the camouflage gives them a better chance to sneak up on prey and capture prey, and therefore more likely to survive, pass on their alleles to the offspring, and that's why the frequency of that allele being able to change color or continued in the population. Anyway, the key points here are camouflage stops you being eaten by predators or allows you to catch more prey. Right, 10.3, scientists concluded that it was probable that disruptive selection was leading to sympatric speciation in the wall gecko. Use the information in the passage to evaluate this conclusion. So just kind of remember that sentence because I'm going to go up to the information. We want to evaluate, is it sympatric or allopatric? Is it disruptive and what evidence is there? And is it speciation? So let's check it out. Right, just underlining some of these. Let's have a look. Well, we've got pale geckos living on walls and we've got dark geckos living mainly on the tree trunks, but they can go on the walls. So
some people would say, hey, it's not sympatric, it's actually allopatric. You might be wondering what allopatric is. Well, allopatric is when you're separated by a geographical barrier. And some people would say, well, living on the walls or living on the trees are two different environments. They are separated, they're not living in the same habitat. So maybe it's a type of allopatric. Although there isn't really a barrier preventing them from getting there. That's a choice that they're not going there. So actually, maybe not allopatric, but certainly we might conclude they're living in different environments, and maybe that's not sympatric. However, the fact that the diurnal geckos can occupy different surfaces during the day, the diurnal, not I don't know what I said, but the fact that they can go on the trees and walls, yes, they mainly live on the trees, but they can go on the walls, means that it is not allopatric and it is sympatric speciation. So I just want to make the point that they actually do live in the same environment because they are able to mix. Now, if they're going to speciate, there needs to be no gene flow, no genes flowing, even though they're living in the same environment and the diurnals can go on the walls where the pales are, they need to not be breeding. So why are they not breeding? They are reproductively isolated. So I want to mention that they must be reproductively isolated, which prevents gene flow between the two species if they're living in the same area. And I could say, what evidence would I say for the sympatric? Well, there's no geographical barrier. There is nothing stopping the diurnal geckos, the dark geckos, meeting with the pale geckos on the walls. Yeah, there's nothing stopping the dark ones going on the walls. We know the pale only live on the walls, but there's nothing stopping the dark mixing with them. So they're not geographically isolated. They're reproductively isolated. They're obviously not reproducing, as we're not getting a mix of those genes. No gene flow. Now, mutations can occur in those two different populations, and based on their environment, you've got natural selection happening differently, which is leading to speciation and leading to the selection of the different skin colors or different colorations. They're different phenotypes. So how do I know? What's the evidence that this is disruptive selection? Because that was part of the evaluation, that sympatric speciation is leading to disruptive selection. And the evidence is that we have two extremes of phenotype. We have pale and we have dark. So two extremes of phenotype. That's the evidence that we have sympatric speciation. And this suggests, this is the evidence that we have disruptive selection. If we had 3 or 4 different phenotypes and we had a mix, then that wouldn't be disruptive. The fact that they're two extremes, i.e. dark and light, is evidence that we have disruptive selection going on. Disruptive selection. Another point is that it did mention diurnal has a distinct genetic group, and that might make us think that they're different species. The diurnal, they're saying their genetics are distinctly different than the nocturnal, the pale geckos. The dark geckos had a distinctly different genetics. However, they follow that sentence up with saying this comparison of the mitochondrial genes also confirmed that all the geckos in the habitat were of the same species. So actually, we are dealing with speciation here. They're still the same species. This is speciation. They're still the same species on their way to separating. They are in the process of speciation. Okay, so we can put all this evidence down here, and this is just a general whiffle, waffle, waffle, which is what an evaluation is about, finding where the evidence is for the scientist's statement. And there's lots of evidence for their statement. I've circled here in green, and all of this evidence in green here is really the stuff that is evidence that it's sympatric and disruptive. I know I can't spell sympatric. I'll put an N there. Sympatric. And then down here we got evidence that it is speciation and they aren't two distinct species already. And there we go. That should get you all the marks for that question there, that 10.3. Now we're going to bus on to 10.4 and 10.5 because this is one of these long ass comprehension questions you get at the end of paper 2 AQA biology. Remember, they're 15 marks. Use your pen to highlight things that you're probably going to talk about when you're first reading the question. Explain how comparison of mitochondrial genes could indicate that nocturnal geckos form a distinct genetic group. Explain how new techniques, I mean modern techniques in the last 50 years, enable the comparison of genes to be completed rapidly. Pause the video, just list off the different genetic techniques you know about for analysis of genes. So,
they're going to have different alleles, right? Because they're in distinct genetic groups or whatever. We want to prove the nocturnals form a distinct genetic group. How are we going to prove that? Well, if they're a distinct genetic group, their alleles are going to be different, or their genes are going to be different. So let's compare their DNA nucleotide base sequence to the diurnals. How are we going to do that? Well, we can use restriction enzymes to obtain fragments. Then we can use PCR to amplify those fragments. We can run them through the gel, electrophoresis, right? Separate the fragments. We could compare the position of their bands. Remember, the lighter bands move further, blah, blah, blah. And the thickness of the bands gives us the intensity of those fragments. We can compare the kind of thickness of the bands and the position of the bands to the ones from the diurnal, or to whatever other comparisons we want to make. We can use DNA probes with radioactive isotopes, etc., or fluorescent ones to identify specific alleles that would be found in the diurnal and the nocturnal, and see if they contain the same alleles. Right, describe and explain two precautions. We could also use the gene machine as well. I forgot about that one. So for this one, describe and explain the two precautions required to ensure, no, let's rewind. 10.5. Describe and explain two precautions required to ensure that the estimate of the size of the population of geckos was valid. Do not include sample size as one of the required precautions. In your answer, include the formula to estimate the size of the population using the mark release recapture method. It's worth 3 marks. Why don't you pause the video, have a think about two things you'd have to do to make the mark recapture method valid, the mark release recapture method valid, and see if you can remember the formula. Let's go. Okay, so the formula is population size that you're estimating. The estimated population size of an area is the number of individuals caught in the first samples. You catch a bunch of lizards, first of all. Then you mark them with a non-toxic substance, like a little marker on them, or you pin something on them. Then you release them again and you catch them again. And you count up the amount you catch the second time after several hours or days, whatever. You release and you catch a second sample. Now you're going to multiply those two factors together. So number caught in the first sample, number caught in the second sample. Then you're going to divide it by the number in the second sample that carried the marker you marked them within the first sample, from the first sample. And there we go. So that will give you the estimation of population size. Now, in order to make this valid, when you mark them, after you've caught the first sample of lizards and you mark them, the marker pen or the pinning device, whatever it is, mustn't endanger them or harm them, because if some of them die during the bit between catching the second sample, it's going to mess up, other than being unethical and cruel to animals, it's going to mess up your estimation of population size. You don't want the marking system to wash off in the rain or fall off when they're running up the trees or up the surface of a wall. And it shouldn't affect their predation. You shouldn't have a glowing bullseye on their back so birds can come and eat them more. That again is going to affect the population size. The other thing is, don't capture the second sample too quickly after you've let them go. You want to let them go and allow them to disperse evenly and spread throughout their natural habitat. If you catch them again too quickly, they might be more easy to catch and you might overestimate the population. So allow a time delay between the first and second sample. There we go. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you lot on the next one.
You're still here? It's over.